scattered through a number of different buildings. The headquarters was in building 11A. I'm not sure of who all was in the laboratory, but I think some of these people were. You can correct me if I'm wrong, but Gideon Schaefer was the research leader at the time. Lowell Owens, Charlie Sloger, Dean Weber, Perry Cregan, David Kirkendall, Freddie Hammerschlag, Dean Kress, maybe a few other people, I don't know. But anyway, this is generally, and then we had a secretary, Jane Wooster. So that's when I first met Tom. And when I was thinking about, wow, that's a long time ago, I didn't realize time was passing quite this quickly. And I went to my files to find some records of this, but they're few and far. I think they were shredded along the tapes. So anyway, Tom and I became friends and colleagues. Then they split us off. This was, we had a frenzied reorganization for about 6 or 7 years ago, 6 or 7 years in a row, and each time I ended up in a different laboratory. So we split off. Tom went over to, I think, the soybean nitrogen fixation laboratory, something like that. I went over to the tissue culture and molecular genetics lab, then the plant tissue culture lab, and then another lab, and kept on moving around. All within a year each time we get reorganized. But then somewhere around 1985 through 1988, we had a plant molecular genetics lab, and I think Tom either moved over with us or somehow or other we became much closer, and we started talking a little bit more about research, things of this nature. And at that time, there was Ram Chandra, Lila Botkin, Gideon Schaefer, Patsy Rhodes, Elliot Herman, a bunch of other people floating around at that time as well. Now, Tom, from my perspective, at least, is an expert soybean geneticist, mapping. He knew a lot about traits, soybean germplasm. He's a bank of knowledge in that area, which I don't have that expertise. So it was a good fit to talk some science and collaborate. I'm more of a molecular biologist type, and I'll show you a little bit of the data and things at the end of what I do. But there's also another common trait that Tom and I have. You may not know this, but he keeps it a secret. He's Irish. And I have the touch of the Irish in me also, or my mother would say the touch of the Blarney, I don't know. But anyway, we have a common bond in that area as well, which is a lot of fun. So Tom has served both as a mentor, giving me support and guidance, insight, and friendship. And then I'd also like to tell you a little bit about the influence he had on my research program as well. And this goes over to soybean cyst nematode. And for those of you who don't know about nematodes, there are these little microscopic worms that live in the ground, or they can live in the ground. In this case, soybean cyst nematode lives in the ground, and it burrows into the root, feeds on the root, saps the strength of the plant, and you get a lower yield. Okay? So it's a major problem in the United States. It causes $1 billion worth of damage. If they would give me just 1 year's worth of that, I'd be very happy in my research program. This is also found worldwide. So it's found in China, Argentina, Brazil, all over the place. So it's a really major problem. If you're going to work on a problem, work on a big problem. So we put together a little program where this is a paper that Tom and I and a postdoc, Jane Weissman, did back in 1992. I didn't think it was that far back either. I thought this was only a few years ago, but it turns out it's further back in distant memory. But Tom and Jane and I sat down and worked out a research program to map one of the major resistance loci of soybean. Soybean can be resistant to one race of soybean cyst nematode or another, but there's a whole population in the ground. So we don't have a plant that's resistant to everything. But if we could learn about how resistance works, maybe we could do something and develop a plant that's resistant to all the different races of nematode. So we developed molecular markers very close to the nematode, or close to the resistance gene. This is important for several different reasons. It puts it gives you a very quick assay, so you don't have to plant plants, add nematodes, wait 30 days, and score them for nematodes growing on them or not growing on them to find out if they're resistance, if they have resistance or not. So you can develop a breeding program much, much quicker this way. This was probably one of the first real applications of having molecular markers in soybean that really targeted a really important gene that we could do something with. This led to developing a map of soybean where we could actually clone out the region that encoded soybean cyst nematode. And we developed markers, and now there are markers today, actually SNP markers that Curry Cricken and other people use that hit right on the spot. But our marker, one of our markers in molecular terms, was almost right there, and it was one of the closest markers that anybody ever came up with until SNP markers became available in the last few years. So this was a really good marker. I didn't realize at the time how important this paper would be, but many people used these markers in soybean breeding. So this started me off. This was, I think, either my first paper or one of my first papers. And now people today are actually testing candidate genes at this locus, at the RSG4 locus. So with that, I kept on going in this particular area. Tom was working in other areas, but this was our collaboration. For me, going through this brought in over $2 million worth of grants over the years just on soybean cyst nematode alone. So it was a very nice collaboration that Tom and I started back 17 years ago or something. So I thank you for that. That was very good. So I'm going to give a little quick overview today now about where this research has gone with soybean cyst nematodes since Tom and I started it back in the early 1990s. To develop resistance in soybean, a question might be asked, what genes are expressed in a resistant cultivar but not in a susceptible cultivar during nematode infection? And maybe some of those genes expressed in the resistant cultivar actually confer resistance, and if we could express them in a susceptible cultivar, then we'd be able to have broader resistance. So we looked at that, and we also have been looking at what genes are expressed in the nematode while feeding that, if we stop those genes from working, might kill the nematode. So we're looking at both from the plant side and from the nematode side. So we use some techniques called microarrays and laser capture microdissection, different techniques to be able to get in and really look at a lot of genes that are expressed in a very small amount of cells. And one of the questions is, what genes are expressed right where the nematode is feeding? Just in those few cells, you have a big root, the nematode's only touching a few cells. What's being done there? So this technique, laser capture microdissection, allows us to go in and look just at those cells. This is a cross-section of a root, and we have the nematode right here. There's a cross-section of the root. This is where the nutrients are coming down the vascular tissue. The nematode is feeding on this cluster of cells right here. So we take a laser and we cut out those cells and we put it in a little cap. And then we go to the next slide, cut, now we have 2, next 3. So we can accumulate just cells that the nematode is feeding on. And then we can find out what genes are being expressed just in those cells without all this other background material that would hide what's going on in those cells. So we asked that question, and we found a bunch of genes that I'm sure you don't care about, but we care about. So we're testing some of these genes in transgenic soybean roots. And we looked both at susceptible and resistant at that feeding site to see what's different about that feeding site, because in the resistant, that feeding site will then collapse and the cells die, and the nematode can't feed because there's no cells there. So if we can figure out how those collapse, maybe we can help broaden resistance to the nematode. Another thing we do is we look at genes expressed in the nematode while the nematode is feeding, and maybe we could turn off some of those genes and kill them. So we have a dye that is taken up by the nematodes if the nematode is dead. So in my lab, we're good at killing nematodes. We can show that, and there's dead nematodes. This is our control. These are live nematodes before I got at them, and there's no fluorescent dye. We can make a molecular DNA construct that can be taken up by the nematode, and when it takes it up, shuts down a nematode gene, and the nematode dies. Okay, so this is targeted against the nematode gene, and we can do a PCR reaction to show that the message for that gene is now gone. Okay, so that seems to work if you do it in a test tube. How about if you do it in a plant? So soybean is hard to regenerate. That means if you transform a single cell, it's hard to get a whole plant back. So that's difficult. So my lab doesn't do anything difficult. We do things simple. So we're not going to regenerate plants. We're just going to make transgenic roots. And to tell that it's a transgenic root, we put in a gene encoding green fluorescent protein. So now we can easily see this root is transformed. Now, if we go down here further, this root here is not green, it's now transformed, so we cut that off. And by doing this, we can encourage this root to grow. Now we have a nice root mass, a bundle of roots, all transformed. We can tell because it fluoresces green. So these roots, along with the green fluorescent protein, we can put in a DNA construct, a gene, or whatever, to test by adding nematodes and say, do the nematodes grow with this gene construct, or do they die on just these roots here? Okay, so we can ask that question, and we've tried a number of constructs. And these are the number of plants we've tested with each of these different constructs. Of course, I'm only showing you our good data. Some genes don't work, but I'm not going to show you them. We seem to, this is the number of females after 30 days that you can count on the outside of a root. Okay? And this is our control plant right here, and we have a lot of nematodes on it because it's susceptible, and so that's all right. And then some of our constructs decrease the number of females on the outside of the root quite substantially. Now, this is not resistance yet. We define resistance by being less than 10% of the control, but it's showing that we're on the right track. It's showing that with some more work, a little more thought, a little more patience and experiments, maybe we can broaden resistance to the soybean nematode. So, Tom, that's where our paper from years ago has brought us today. We still have a lot more work, so come on back every so often and help me out. Tom, as I said, has been my mentor, my friend, my colleague. However, we do have a difference in our strategies. I want to show you this. Tom thinks big, I guess, and these are his big soybean plants. In the laboratory, we don't have enough greenhouse space to have a lot of these. Tom has the whole earth to plant outside. So this is mine here. This is probably pretty much the scale. These are about 6 or 8 inches tall. This is Mini-Max versus Tom's variety. But we come from different points of view, but we still get to where we need to go. So this is an old Irish limerick. You can read it on your own if you want. Many of you probably have seen it before in the old literature. But anyway, I want to thank you, Tom, very much for being my friend and mentor, and I've enjoyed our interactions, and I hope it continues.